another today's lesson of ours. Welcome to another today's lesson, another beautiful lesson of proteins and enzymes and chemicals of life at large. And we have really covered, and we have covered enough as far as chemicals of life is concerned, where we are able to talk about the inorganic nutrients, water, stuff like that. Then the organic, where we talked about carbohydrates, lipids, proteins, and you must know all those ones. And then today we are on enzymes. Okay? Yeah, we are going to do a brief overview of enzymes, and we see what enzymes are and how these enzymes are important in our bodies. Feel free to drop your questions in the chat box. In case the question is immediate, you can unmute, okay, and ask for this question, and ask your question. I will attend to it immediately. By the way, feel free to subscribe also to our channel. It is really good, it is really key. Okay? Yes. So we have talked about proteins, and let's simply talk about enzymes and see what enzymes are. Because you all know that enzymes, they are also proteins in nature. Okay? They are proteins. So let's see what these proteins, enzymes, are. So if you can have a look here, you can see this is our enzyme, and I managed. I liked this and I clipped it. So this is enzyme alpha amylase, which is responsible for germination in seeds. Okay? For a seed to germinate, then this enzyme must catalyze the hydrolysis of starch to maltose in the endosperm, and then the maltose hydrolyzed to glucose. Germination occurs. Yeah, the process of germination. You can review one of the videos of, is it coordination? Yeah, I talked about it briefly. So this enzyme, so important. And so that brings us to the importance of enzymes in nature. These enzymes are really important. Okay? In nature. So before we go far, you need to know what an enzyme is. It might not be new, it might not be old, but we need to know what enzymes are, or what an enzyme is. First of all, an enzyme is an organic catalyst. Okay? We have different types of catalysts. We have inorganic catalysts. Okay? Inorganic catalysts. For example, let me take you back to senior 2. Okay? You remember when you are preparing oxygen? Yes, you just add hydrogen peroxide, okay, to a flask containing manganese oxide. So this manganese oxide acts as a catalyst, but this is inorganic. So also these enzymes, they are also catalysts, but they are organic catalysts. And what do catalysts do? We are going to see. So these enzymes, they are organic catalysts. They are proteins in nature. Yes. In other words, they are made up of a sequence of amino acids, okay, to form a polypeptide chain. And then this polypeptide chain is modeled, okay, is modified to give that 3D shape, okay, that is going to be responsible for catalysis. Okay? The 3D shape, this 3D shape is the one that will be responsible for catalysis. And so they are proteins in nature. So what do? They speed up the rate of a chemical reaction in the body. And what does that mean? Naturally, These chemical reactions are spontaneous. They can occur, okay, on their own. That's what spontaneous means. You eat your potato, you eat your cassava. The cassava can digest or can be digested without enzymes. You eat your matoke, the matoke can be digested even without enzymes in our bodies, and we get the energy out of them. But that spontaneous reaction is slow, that you might eat cassava today and you get the nutrients on 25th December. So, how would you survive within that period from now up to 25th December? So what these enzymes do, they do increase the rate of a chemical reaction, okay, by providing an alternative path or by lowering the activation energy, anyway, of such a chemical reaction. We are going to see. So they speed up the rate of a chemical reaction in the body, and finally they remain unchanged at the end of a chemical reaction, such that they can catalyze further reactions. So that's why enzymes, they are effective even in minute quantities, even in small quantities. These enzymes will still do carry out their catalysis. A job that is done by 1,000 enzymes, the same job can be done by 10 enzymes. However, now what is going to matter is the time. If 1,000 enzymes take 1 hour, 10 enzymes will take 10 hours. Oh, 1,000 divided by 10 times whatever. You studied proportions, you can find the proportion handy. But then 1,000 enzymes will take less time and 10 enzymes will take much time. But however, the work will be done. Okay? Yes. So enzymes remain unchanged at the end of a chemical reaction. They are not reactants. They just participate in a chemical reaction as catalysts. And catalysts remain unchanged. So, think about this. You wake up one day and you don't have enzymes. Boom! Do you happy? How do you feel? Do you feel good? Are these enzymes really important in our bodies? Yes, they do. Like how we have seen is that these enzymes, they do speed up the rate of a chemical reactions. Like I told you, all these chemical reactions, they are spontaneous in nature, in that they can still occur on their own. If you get an iron bar and you leave it outside, doesn't it rust? Yes, it does. That reaction is spontaneous. Okay? If you get, think about other reactions. Okay? Yes, however, others, they may not occur spontaneously. For example, if you expose matchbox and oxygen, or if you expose the carbon, the charcoal, and oxygen, they might not occur until when you light. But most reactions do occur spontaneously on their own. However, now, the rate at which these chemical reactions will take place on their own is going to be so slow that they cannot sustain your demands. For example, imagine you eat your potato today, or you eat your posho today, and you get glucose out of it after 1 year. after 1 month. Think about it. Okay? You eat your rice today and you get your glucose after 1 month. How are you going to be surviving in this period? Even after getting the glucose, now to get the energy out of glucose, it also takes another year. Man. Even to use the energy itself. Man. So you can imagine how easily, how hard life would be if it's there even. If it would be there even. Okay? So as an evolutionary trend, or as an evolutionary strategy, then organisms have evolved tiny organic molecules that are proteins in nature to speed up the rate of a chemical reaction without changing the product or the nature of the chemical reaction. In other words, you don't interfere with the products and the nature. If the reaction is to take place forwards, or if the reaction is exothermic, for instance, then the reaction remains exothermic. If the reaction is endothermic, in other words, it remains endothermic. And those are the reactions that take place in our bodies. For example, for you to form ATP, then energy must be absorbed. So that when you break down the ATP again, then the other energy is released. And you are able to harvest, you are able to use the energy in your body. So that would happen on its own, but these enzymes speed up the rate at which that reaction is going to occur. How do they do it? By lowering the activation energy of a given reaction. Now somebody must be asking, what is the activation energy? If I try to sketch something of this kind, okay, trying to sketch, so this one will be the reaction path or the reaction coordinate, for instance. Okay, path, and then this one is going to be the potential energy. So for a reaction to occur, so from this graph, we shall see that this one is the measure, the measure of energy, okay? And you may have your reactant molecule at this energy. Oh, these are your reactants, okay? Now the reactants, for them to react, okay, to form a product, they must first undergo some collisions, and if the collisions are built enough, then they will increase, their energy is going to increase due to those collisions, okay, and they form an activated complex, okay, activated complex, okay. Now this activated complex, it is the one that will dissociate to give us the products. Now the energy required, okay, to raise these reactants, okay, to obtain the energy of this activated complex is what we call the activation energy, okay, activation energy. Now for a reaction to proceed forward, for a reaction to be feasible, for a reaction to be possible, then the reactant molecules must collide with each other, that is if they are in proper orientation. If this is a reactant molecule and this is another reactant molecule, the reactant molecules must collide with each other. In the process of collision, energy is built up. Okay? Energy is built up in the process of collision. That if this energy builds up above the activation energy of this reaction, then you'll be able to form this activated complex, which will dissociate to give you the products. And the higher the activation energy, the higher the energy needed by these reactants. So the higher the collisions you can imagine for a reaction to proceed. But if the energy built up during this collision is not sufficient enough to outweigh or to overcome the activation energy, then the activated complex between the two reactant molecules formed, it will dissociate back to give you the reactants, and you won't be able to form the products. So in case the activation energy is higher than the energy built up in these collisions, then the reactant molecules will dissociate to give you back. So the activation energy will dissociate to give you back the reactant molecules. Not so? Yes. Now, what enzymes do if the reaction is spontaneous? I mean, if the reaction occurs on its own without enzymes, the activation energy will be so high that it won't easily be achieved. That the number of products formed per minute is so small. The number of products is going to be small. The products will be very few, too small to sustain you. But what enzymes do, and not just enzymes, but catalysts, what catalysts do, they are going to provide an alternative path. Okay? For example, in organic catalysts, they provide that surface where the reactant molecules will get, they will be adsorbed on the surface of the catalyst, and when they are adsorbed on the surface, then the activation energy is lowered so that you easily, the activated complex easily dissociates to give the products. And also what enzymes do, now enzymes, they have different mechanisms of catalysis. Some do so by proximity, by bringing the reactant molecules close to each other. Others do so by forming temporary bonds. There are different theories that try to explain catalysis. Okay? You form temporary bonds so that the reactant molecules can now easily react with each other to form products. Others do so by straining the reactant molecule is put into an enzyme, and then the enzyme strains the bonds. And when you strain the bond, then you easily break the bond. So there are different mechanisms of catalysis. Okay? So by doing so, you are lowering the activation energy. This one, the activation energy for such a reaction, so that you attain the products so easily, hence increasing the rate of reaction. The number of products formed per minute is going to increase in an enzyme controlled reaction than when the reaction is spontaneous. So basically that's how simple this concept is. Okay? Yes. So they also control metabolic processes, hence promoting normal body functioning. This is okay. All metabolic processes, all body metabolism is performed by this enzyme. So without enzymes, without enzymes, then man, I don't know how we will survive. So let me give you some reading assignment. You can go to Google and read about the Michaelis Mountain kinetics. This is for your own understanding. I think it's E. This is for your own understanding. Nobody is going to ask you this, but you can read about it to expound your knowledge as far as enzyme catalysis is concerned. But don't do those calculations. Don't do the calculations. That's another chemistry topic. Just read through to appreciate. Okay? So having that simple understanding, not simple but basic understanding of enzyme activity, let's actually see some theories that are put forward to explain the enzyme activity. The theories that are put forward to explain how enzymes actually do carry out catalysis. Okay? Well, let's talk about enzyme action. Now, the enzymes have a unique surface, okay? And I'm quite sure you talked about this or you've ever heard of this. So they have a unique surface. Not the whole enzyme actually carries out catalysis. Not the whole of it. But rather, some unique surface on the enzyme that is responsible for catalysis. Okay? And that site is called the active site. Now, it is the site that is responsible for catalysis. So it's where molecules that do adsorb, or it's where molecules that do attach on the active site on the enzyme, and then they form the enzyme substrate complex. Okay? And it's now this complex that will dissociate to give you the products and your enzyme back. And the enzyme remains unchanged. Okay? When finally this reactant molecule or reactant molecules attach on the enzyme, they will remain in close contact. That is infinite contact. Okay? They will remain in close contact until when the enzyme dissociate, until when the products, if it is breaking down the products, then until when your one simple compound breaks down to give the two products. Or if it is formation of a bond between two reactant molecules, then they will form a bond, and when they form a product, then it will detach. Let's have some simple illustration here. Okay? So it's like this. You have your reactant molecules, or you have your enzyme, okay? But not the whole of this enzyme can carry out catalysis. But some site, okay? Certain surface on the enzyme with some specific configuration or shape, or some specific configuration or shape, it is the one that is responsible for catalysis. And this site here is unique to this enzyme, and it will be unique to another enzyme B. Okay? So you may find now this enzyme B has such a shape, and the whole of this is the site. The whole of this is the site. And this is enzyme B. And it is this site that is responsible for catalysis. Whereby Now this site will bind with your substrate. Maybe my substrate is something like this. And the site also should have a configurational shape. Did I say configurational? I wanted to say complementary shape. The shape must complement. Now, there is a mistake students usually make. Instead of saying complementary shape, they say substrate have same shape. This is wrong. There's a difference between same and complement. Let me use what you like most. When you are choosing for your partner, you look for a partner that complements you, not the same as you. Boom. You look for a partner that complements you, not the same partner as you. You cannot find somebody who is exactly like you. You cannot. But somebody who complements you. Okay? That means if you are a drunkard, then you don't choose for a drunkard. You look for somebody who complements you. Maybe not drunkard. If you are short, then don't look for somebody who is short. Look for somebody who complements you. Maybe tall. Okay? So complementing doesn't mean same. Okay? But fits. For instance, use padlock. The key complements the padlock. But do they have the same shape? If you try to open inside where the key goes, is the shape the same? You may find if the key is like this, let me use FDC's key. Okay? And that's FDC's key. So you may find if the key is like this, the inside of a padlock cannot be like this. Cannot be like this very shape. At least it will be kinda like this. Okay? Now are these the same? Are these shapes the same? They are not the same. But these shapes do complement each other. Whereby if you bring this one here, it will fit. So that's the difference between complementarity. Okay? Yes, and same. So if you write same, I will cross you. And I've encountered many students writing same. Enzymes have a specific shape and substrate have a same shape where that fits. And you wonder, it's not same. It's not same, but rather complement. So the substrate they have a specific shape that complements the shape of the active site of the enzyme. In that when the two, obviously they will be in space and they will have to collide with each other. And when the two collide with each other, then the substrate, it will fit in the active site and it will form an enzyme substrate complex. Okay? An enzyme substrate, and this is your substrate now. Okay? Something of this kind. And this is your substrate. Now this is the enzyme substrate complex. Now that enzyme together with the substrate, they will remain in close proximity. They will remain bound to each other, and probably the enzyme will strain. We shall study the theories. This enzyme will strain the bonds that are formed within these substrate molecules, probably, depending. And when you strain, you will create tension on those bonds and they will end up easily breaking down. And when the bonds are broken down, then you form product A. Okay? and B. And you form back your enzyme. Now, when the product A and B are formed, now these temporary bonds that are formed between the enzyme and the substrate, they will weaken. They will no longer be there. So that now the product molecules will easily detach from the enzyme, exposing, okay, the active site again for another substrate molecule to bind. And the enzyme will remain unchanged. Not so, but you have your products. Now, when this, okay, I'll explain that. I was going to talk about an inhibitor. Now, for instance, this one, it can be, let's say maltose. Okay, let's say sucrose or maltose, same, they are all disaccharides. Sucrose, okay, it's a disaccharide. It will attach in enzyme sucrase here. And then the enzyme sucrase will strain the 1,4-glycosidic bond between the sucrose, between the fructose and the glucose in sucrose. And when you strain that bond, you add their water molecule, and boom, the bond will break down, and you form the two, the glucose molecule here and the sucrose molecule here. So there's a fructose molecule there, and you have your enzyme back, and the enzyme will be waiting for another substrate molecule. So it's as simple as that. If you understand this basic, then trust me, everything is gonna be cool. Any queries for me? Yeah, so thanks for that wonderful question. Now let's see how we can classify enzymes. This is some simple, and it is just simple science. Now, we classify enzymes basing on the type of reaction that an enzyme catalyzes. And so those are the different classes of enzymes that we have. For example, if the enzyme catalyzes the reaction which involves both oxidation and reduction, then that enzyme is categorized under oxidoreductases. For example, we have enzymes that are involved in respiration. Most of them are oxidoreductases. Okay? They do catalyze. Now, an oxidation reduction reaction, it is a reaction that occurs where one compound is oxidized and another compound is reduced. For instance, you can have a look at this. For example, you have a reaction where you have the chlorine gas and you are bubbling chlorine gas. Let me use some basic that we all know. In a solution of bromide ions, for instance. And here we shall get the chloride ions, okay, and bromine, and the 2 here. So if you are to balance, we shall put a 2 here and then a 2 here. Now this is a redox reaction. This is an oxidation reduction reaction or equation, whereby you can see in this the chlorine is being reduced. You see, from 0 oxidation state to -1 oxidation state, there is a decrease in the oxidation state. So chlorine is reduced. But now for bromine, here it is in -1, but here it is in 0 oxidation state. Now from -1 to 0, there is an increase. So the bromine is oxidized. So this is a redox reaction, an oxidation reduction equation or reaction. Now let's say there is an enzyme Z or enzyme A that catalyzes this reaction then this enzyme is going to be categorized, it's going to be classified as an oxidoreductase. Okay? For example, dehydrogenases. Okay? Dehydrogenase, oxidase, or reductase. Now, in in respiration, they usually undergo oxidation, where one compound is going to be oxidized and another compound is reduced. Okay? One compound is oxidized, another compound is reduced. For example, in the electron we haven't studied respiration, but I made videos. They are there in our classroom. You can go there and check. You can go and watch. Okay? And enjoy the science. So that by the time we reach there, for you are kawa. You have all, you are doing revision, which is good. So now these enzymes, they do catalyze reactions which involve removal of hydrogen and carbon dioxide, whereby in one compound where you've removed hydrogen, you have oxidized it. Okay? Then another compound, you add there electrons, or you remove hydrogen together with electrons. Then the compound that receives the electrons, okay, together with the hydrogens, the protons, it is reduced. Even in electron transport system, ETS, there are electrons that flow where one compound is oxidized, another compound is reduced. So such enzymes that do catalyze redox reactions are referred to as, or they can be categorized as, oxidoreductases. Then we have transferases. Now these ones, they do catalyze reactions which involve transfer of a given group, of a functional group, from one compound to another compound. Transfer of a functional group from one compound to another compound. For example, during urine formation or during deamination, for instance, it is the same. You know that your amino acids, they have the amino group. Okay? This is going to be your amino acid. Okay, let me use this shape. So this is going to be our R group. Okay? You know the other group. And then this is your alpha carbon. And this is the amino group. Okay? I've just changed it. And then this is the COO. Okay? H. So this is our amino acid. Now, our bodies don't store proteins, for instance. You know that. So what our bodies do, they are going to deaminate. They are going to remove this amino group from the amino acid and then bring the keto group, this O with a double bond, and then they insert it there. Then they convert this ammonia into urea by adding it to a methyl group, and then you add and remove water at the same time. You form, the ornithine cycle. You can have a glimpse at it. And so now, if you add the keto group here, this is the new compound that will be formed. And you can see that clearly this is a carbohydrate. So you have C and then O and then COOH. So this is really a carbohydrate. Don't you see this? Whereby you can now feed this in the Krebs cycle and you get energy out of it. So the enzyme that does that is a transferase. Okay? Because it involves transfer of a given group. Even during phosphorylation, you transfer a phosphate group from a high energy compound to form another compound. For example, during formation of ATP, we have different ways whereby you can form ATP by substrate level phosphorylation, or you can form ATP. Okay, for instance, during phosphorylation, you transfer a phosphate group from ATP and you add it to another compound, for example, glucose. Or during formation of ATP by substrate level phosphorylation, you transfer a phosphate group from one compound to another compound to form to ADP, and you form ATP. Okay? So the enzyme that does that is a phosphorylase because it involves the transfer of. Then hydrolase, hydro comes from water. So reactions which involve addition of water or loss of a water molecule. Those ones, they are hydrolases. And these are the common enzymes. These ones, common enzymes, almost all. For example, we have proteases, we have lipases, starch, amylase, almost all enzymes. Then we have lyase. To lyase means to break, to split. Lysis comes from lysis. Lysis means splitting without use of water, for instance. Okay? It's like you get a panga or an axe and you split. Okay? So that is lysis without using. So it's not a condensation reaction, in other words. And enzymes that do that, they are lyases. And for example, we have decarboxylases and carboxylases. Decarboxylases, they do remove carbon dioxide. Then carboxylases, they do add carbon dioxide. So now these ones, they do catalyze breakdown of chemical bonds without addition of water or a new group to the free bond. Then we have isomerases. These ones, they do catalyze isomeric reactions that involve rearrangement of atoms to form a new compound. Okay? Without loss or gain. You rearrange, okay, to form a new compound. For example, in respiration, you find there enzymes. You find that after phosphorylating the glucose, anyway, let me talk about it. The first step in respiration is phosphorylation of the glucose molecule to form glucose 6-phosphate. Now the glucose 6-phosphate that is formed, it will undergo isomerism. It will rearrange itself to form another compound, a new compound, which is fructose 6-phosphate. Fructose 6-phosphate. You study this in respiration, or you can go and watch the video and enjoy. So fructose 6-phosphate. Now there is nothing has changed here. All these two, they totally have same. Now here I'm using same molecular formula, same molecular mass, but a different structure. Okay, so that is isomerism. So now the enzyme that is responsible for this, it is an isomerase. Okay, it is an isomerase. Okay, and without this enzyme, then this cannot occur. And if you cannot convert from glucose to fructose, then it will be hard for you to add another phosphate group so that you form fructose 1,6-bisphosphate. It becomes hard, if not impossible even. it becomes hard. Okay? Yes. Then we have the gases. Now these ones, they do add two energies, or they do catalyze the formation of a bond using energy that is derived from ATP. E.g. in DNA replication, we shall find there these enzymes, that are gases. So basically, that's how we can classify these enzymes, basing on the type of a chemical reaction an enzyme performs. Then nomenclature, it's easy. We name these enzymes by adding suffix ase, ase, on the stem name of an enzyme, on the substrate, not stem name. This is not organic chemistry. On the substrate that enzyme catalyzes. For example, if you have enzyme and the substrate, if you have enzyme peptidase, then the substrate must be a peptide. If you have enzyme lipase, then the substrate must be a lipid. If you have maltase, so just ASE, ASE. This is basically something simple. Cellulase, this is supposed to be cellulase, okay? Then the substrate must be cellulose. However, there are enzymes which have retained their trivial names, which don't conform to this nomenclature. For example, amylase. Amylase, it's not starches. If you want to continue with this nomenclature, then it will become starches, but it's not starches. Okay? It remains amylase. All proteases, by the way, apart from peptides, peptidases. But anyway, this is not a protein, it's a peptide chain. So all proteases, okay? You can talk about pepsin, you can talk about trypsin, you can talk about renin. Renin. Is it double N? You can fact check this. Renin. All proteases, they are not called, no, it is pepsin, it is trypsin, it's renin. Okay? Not. So this is just a general term that tries to generalize that. Okay? Yes. Any queries before we talk about the rock and key hypothesis? Yeah, since there are no questions, then yeah, it's okay. Now, like I told you, science, it is people do research and then come up with conclusions. So we are studying all this. Can we try? There are theories that do try to explain how enzyme activity, how actually enzymes do carry out this catalysis. And they are still hypotheses, by the way, not yet proven. They are still hypotheses. Okay? Yes. So we have two hypotheses that try to hypothesize, that try to explain. It is just trying to explain how enzyme catalysis occurs. And the first one is the rock and key hypothesis, and the second one is the induced fit hypothesis. Now, with the rock and key hypothesis, this one is just simple. As its name suggests, that is Katorva's English. As its name suggests, this is the rock and key. You know the rock and you know the key. So now this theory assumes the enzyme to be a rock and then the substrate to be a key. Okay? It assumes the enzyme to be a rock and the substrate to be a key. And therefore, for catalysis to occur, then the key must fit in a rock, just like you use your key to open your padlock. If you misplace your key, you cannot use your neighbor's key. You cannot use your friend's key. You must use your key. And the key has some specific configurational shape that fits the padlock, that complements the shape of the padlock. Okay? So that now when you insert your key, then the padlock, the two will complement, will fit each other. And when you try to rotate, then the padlock will open. So the same applies to this theory. It assumes the enzyme to be a key, a padlock, and the substrate to be a key. And the two, the enzyme having a specific shape, okay? It has a specific shape of the active site, the enzyme, that a substrate must complement. Now, most of you will say, same, I will cross you. That a substrate must therefore complement and fit into. Okay? Now, after fitting into, then you form your enzyme substrate complex that will dissociate to give you the products. And the key, and the substrate, and the enzyme, which is the rock, it will remain uninterrupted with. It will remain unchanged and ready for catalysis to catalyze other reaction. So that's the rock and key hypothesis. So you can have some illustrations here. You can have a rock. Okay? You can have a rock. This is your enzyme. Okay? And then this is the active site with some specific shape. Specific. And by the way, this shape is rigid. It doesn't change at all, according to this hypothesis. It does not change at all. It's just there, like boom. And then the substrate is also there. Then the two come, boom, bind. And when they bind, they're going to form the enzyme substrate complex. Specific. Specific. No other substrate can bind there unless it has a specific shape or it has a complementary shape that complements the active site. So now after binding, then you form an enzyme substrate complex which will now either dissociate or combine, like how you see this, to form a product. And now the product formed, it will have a different shape that can no longer fit into the active site. And so what will happen? Then it will detach away from the active site and you have the free enzyme and the product ready for another catalysis. That's the rook and key hypothesis. And this theory is, by the way, good. Real good, but man, it has some challenges. So you can also have this. You have a specific shape. This is your enzyme. You attach in a specific manner. Specific. And then after touching, you strain a bit. After straining, you form the enzyme substrate comp and break, you form the products. Okay? That's brilliant. So this theory is good. It is really good. Okay? Why good? Number one, it explains the specificity of the enzyme. Why enzyme A cannot catalyze the reaction for enzyme B. And it's really good. That's how it is in nature. It is what it is. That's how it is. Okay? Enzyme A cannot catalyze reactions for enzyme B. Why? Because they totally have different shapes. Okay? The two enzymes have different shapes of the active site. If, for instance, this is enzyme A and this is the shape of the active site, then enzyme B must be different, or it will be different. And these enzymes have different active sites, like how you can see, how you can glimpse at this. And because of the differences in the active sites, then the substrate molecules must therefore have totally different shapes. Maybe this one will be kinda like this. Totally different shapes. And this one will be kinda like this. Totally different shapes. And this one cannot fit into this, and this one cannot fit into this. So this will be specific for this, and this enzyme too will be specific for this substrate. And it's really key as far as rock and key hypothesis is concerned. It's really good. Okay? Good. It tries to explain why an enzyme can be used over and over and over again. An enzyme can catalyze different reactions. We have seen that after you form the enzymes, after you form the enzyme substrate complex, okay? Maybe this is the enzyme and this is the substrate. After forming this enzyme substrate complex, the complex will strain, the enzyme will strain the bonds into the substrate, and boom, this reactant molecule will break and you form your two products. And the enzyme formed, it will remain unchanged. The shape of the active site will remain unchanged, the same. Okay? And if the shape remains, then what does that imply? Another substrate can bind, okay, to the active site. And if it binds, the same thing happens. And another substrate, and another substrate, and another substrate. So this is real good. Okay? This is real good. It tries to also explain why the rate of enzyme controlled reaction is limited by increasing the substrate concentration, or is affected by the substrate. And we shall see factors which affect enzyme activity. And we shall talk about substrate concentration. But when the number of substrate molecules increase, then at any time t, all enzymes will be engaged. And so the rate is going to increase. But further increase does not guarantee that the reaction is going to increase. It will become constant. Why? Because all enzymes will be saturated at any time t. All active sites will be saturated. All enzymes will be active. We shall try to explain these factors, okay? And see how. Number 3 or number 4. It tries to explain how the enzymes can be reduced or can reduce the activation energy of a normal reaction by showing that when a substrate binds to the active site, the substrate molecules become slightly stretched, which strains the bond, and so less energy is needed to break the bond. However, now it is the induced fit that tries to explain this so much. But also this one also talks about it. Okay, that when the substrate binds to the enzyme, then the substrate is brought into proximity. That's not the straining. It's not valid here, but it brings the two into proximity, and when they are close together, then they can easily react with each other, hence forming the products. So by bringing the two reactant molecules close together, you are lowering, okay, the activation energy. You are bringing the two reactant molecules in proper orientations so that the collisions between the two will be enough to build activation energy to form the products. Okay? Yes. But the straining, the stretching, this is not of an arrogant. We shall find it down. I'm going to edit this. I'm going to change this. Then, finally, is it finally? How and why enzymes can be inhibited? For example, competitive inhibition. Yes, you have the same shape as the substrate. So you can attach to the substrate. You can attach to the active site and you prevent. But now here when you attach, then you won't dissociate from, you won't detach from the active site. You kuremeramu. So when you don't detach, then there is no way how that enzyme will combine with the substrate molecule. But still you cannot dissociate. So you inhibit the reaction. How enzymes can be inactivated by high temperature? We shall see factors, but high temperatures do the nature. The enzymes and they lose their 3D shape. And when they lose their 3D shape, then they lose the active site and so they cannot catalyze. So basically that's where we shall stop. Let's do some reading about the induced feed. And before the induced feed, I want you to make research the limitations of the rock and key hypothesis. Why the rock and key hypothesis is still rejected. Okay? So that by the next lesson, you'll be covered. Limitations, okay, of the rock and key hypothesis. So go and make research, do some reading on this. Okay? Yes. Then you come, this is where we shall begin in our next lesson. Otherwise, guys, have a nice time. Don't forget to subscribe, by the way. Don't forget. Just hit the subscription button.